start with the brain development. Okay, your brain develops as the neural tube. Okay, the upper part of the neural tube effectively becomes the brain, and the rest becomes the spinal cord. So what you need to know here, you need to know and identify primary brain vesicles. You need to know secondary brain vesicles. And I will ask you, what do secondary brain vesicles develop into? Okay? So you can expect the question, myelencephalon. What does it develop into? It develops into medulla oblongata. Okay? Now, if you look at the image of the brain on the right, you can see that brain grows faster than the cranium, and it means that the brain has to fold over itself. Okay? Does that make sense? It folds over itself. Forming this folds, and turns out that these folds on the surface of the brain, called gyri and sulci, are very important for the proper brain function, because folds mean that there is an increased surface area, and increased surface area means that there are more neurons on that surface. Turns out that these surface neurons are the ones that are involved in our conscious thinking. So brain is pretty squishy, and it has to be protected by some kind of a cushion, some kind of, let's call it an insulation. So the cushioning role is taken up by the cerebrospinal fluid. Cerebrospinal fluid is produced in the choroid plexuses, shown here at the bottom. In the choroid plexuses, ependymal cells form a barrier between the blood, the capillary, and the cavity of so-called brain ventricles. So this image shows you lateral ventricles that are located on the sides of the brain. The third ventricle and the fourth ventricle. Third and fourth ventricles are connected by the cerebral aqueduct, and lateral ventricles and the third ventricle are connected by interventricular foramen. Fourth ventricle, in turn, drains into the central canal of the spinal cord. Now, if you would look at the image on the bottom, you can see that the cerebrospinal fluid that surrounds and protects the brain is pretty much all located, not pretty much, but all located into the subarachnoid space. One may wonder how thick the layer of cerebrospinal fluid is. Its thickness is around 1 penny. Just 1 penny. It's super thin. Okay? So what comprises? What are the components of the cerebrospinal fluid. So first of all, it's water, of course. Cerebrospinal fluid is fairly similar to plasma in its composition. However, cerebrospinal fluid, just FYI, because you're going to be looking at the data for I'm blanking on the word. Data of analysis for patients, right? Cerebrospinal fluid should contain very few cells, very small number of cells, and should not contain any infectious agents, obviously. So any inflammation, any infection of the brain or meninges usually is reflected by the presence of various blood cells, mostly white blood cells, in the cerebrospinal fluid. Okay? So you can see that since cerebrospinal fluid floats the brain, it basically reduces its weight by approximately 97%. Your brain is literally floating in the water. So we already said that cerebrospinal fluid is formed by choroid plexuses in all four ventricles. Turns out that daily choroid plexuses produce 500 milliliters of CSF. However, it's important to understand that the total volume of cerebrospinal fluid that humans carry is only 150 mils. So it means that somehow cerebrospinal fluid has to drain. So where does it drain through? It drains through arachnoid villi. Arachnoid villi, you can see them. Say again? Yes. Subarachnoid villi, arachnoid villi are the structures that drain cerebrospinal fluid. So this is one villus, another villus. So basically, what I want you to appreciate is that we have a faucet that produces the fluid, and we have the drainage that removes the fluid from the subarachnoid space back into the blood. If there is some kind of an obstruction of an arachnoid villus, cerebrospinal fluid is going to accumulate in the brain, not in the brain, but around the brain. And this condition is called hydrocephalus. Now, hydrocephalus, think about what happens. When cerebrospinal fluid accumulates, right, it starts exerting pressure on the brain. So, newborns tolerate hydrocephalus better than adults. Can someone propose why? No, not. Yeah. Yes, so they have fontanelles. Their skulls are not completely ossified. So there are soft connective tissue spaces called fontanelles. Okay? Does that make sense? And the skull can sort of give in to the pressure, reducing the pressure that actually is exerted on the brain. Now, in addition to cerebrospinal fluid, another structure that protects and secures the brain is meninges. So, meninges are membranes formed by connective tissue and they are superficial to the brain. So let's take a look at the structure of meninges right here. I'm going to kind of, forget about the skin of the scalp and, the bone. Okay, so this is bone. Now, the layer that clings directly to the bones of the skull, It's called dura mater. It is the thickest, the hardest, the strongest layer, the strongest meningeal layer, which consists of two sublayers. The layer that sticks to the bone is called periosteal. The layer that clings towards the brain is meningeal. On this image, you can see periosteal layer and meningeal layer. You can see that sometimes periosteal and meningeal layer separate. When they do that, when they separate, the space between them is occupied by the blood vessel called a sinus. This is a type of a vein found exclusively in the brain. You see that it is shaped not, it's not round like a vein, but more of a triangular structure. But other than that, it's a typical vein that drains the blood away from the brain. Deeper to dura mater is arachnoid matter. It's called arachnoid because it is attached to the deepest layer of meninges, to the pia mater. It is attached to it through this web-like projections, web-like fibers. So the deepest layer is pia mater. Pia mater is a very thin and delicate connective tissue which is attached directly to the brain tissue. What I want you to really pay attention to here is that there is a space between arachnoid and pia mater. The space that's right here. This is the space where cerebrospinal fluid circulates, and also this is the space in which blood vessels that supply brain with the blood and drain blood from the brain are located. Why is it clinically relevant? If somebody has a head trauma, and as a result of that head trauma, one of those blood vessels that are shown here gets popped, the bleeding is confined to the subarachnoid space, and we call this subarachnoid bleeding. This is pretty dangerous because blood will exert pressure on the brain, disrupting its function. And the problem with subarachnoid bleed, it's often hard to diagnose. And there are examples of people who felt pretty much fine and passed away a few hours later because the bleeding developed so fast. Now, in addition to wrapping the brain up, another function of meninges, specifically dura mater, is to secure the brain within the skull. For instance, here in this image, you can see various parts of dura mater, such as falx cerebri, tentorium cerebelli, and falx cerebelli, that partition the brain and secure it to the skull. On the macro level, you can clearly see the brain is organized into hemispheres, left and right. So this is the left hemisphere, this is the right hemisphere. Hemispheres are separated by the median longitudinal fissure, and on the surface you can see that there are ridges called gyri. For example, this is the post-central gyrus. And gyri are separated by sulci. For example, this is the central sulcus. By themselves, gyri and sulci don't have any functional relevance, but they're used to identify different lobes of the brain. So, brain is divided into 5 lobes. The frontal, located in the front. The parietal lobe, located on top of the brain. The occipital lobe, which is in the back, and the temporal lobe, which is on the side. These 4 lobes, and the 5th lobe is insula, and it is deep within the brain, so you really cannot see the insula lobe on that illustration, and I can guarantee you that in anatomy, insula lobe will not be the part of your anatomy exam. Okay? Now, do you need to know where the lobes are? The answer is absolutely yes. Do you need to know which structures separate the lobes? In other words, can I ask you, what separates frontal and parietal lobe? I cannot. I'm not going to ask you that. It's a very specific knowledge, and unless you plan on working in like neuroanatomy, you don't need this. Yes. So again, front of the hemisphere is the frontal lobe, back of the hemisphere is occipital lobe, the side is temporal, the top is parietal, and the very deep is insula. Okay? So our first topic of conversation would be the cerebrum, which consists of 3 major parts. Cortex, the superficial layer, which we sometimes call gray matter, which is not entirely correct. Cortex contains all centers responsible for any of the conscious activities or perceptions that we may have. In other words, your entire conscious life is within the cortex. Deeper to the cortex is white matter. So white matter is white due to the presence of myelin, vast presence of myelin. And myelin, as you may remember, covers axons of neurons. The role of axons is communication. So, whenever you see white matter, you are looking at the structures that allow different parts of the brain to communicate between each other. Now, deep to the white matter in cerebrum is basal nuclei. Basal nuclei is responsible for the control of movement, and it's not under our voluntary purview. I'm going to give you a highlight of the cortex. So this is the cortex. This is the white matter. It's not only white matter, this is white matter too. And these are the parts of basal nuclei. So it's like a sandwich. Gray matter on the outside, then white matter, then basal nuclei, which are gray matter on the inside again. Okay? Reasonable? What I want you... Question? Can't hear you. Yes. And I can see. Okay, yes, I can. So outside layer, which is a gray matter, this layer, the outside layer, that's the cortex. Okay? This is our thinking, you know. The white matter under it, that's a communication. And basal nuclei, like this, that's the control of movement. Okay? Now, how do we know that? Uh-huh.
Did you say that's the control of all movement or just involuntary?
Involuntary. Okay, let me rephrase it. This is the level of control of movement that we can't consciously appreciate.
Okay.
So, there are structures in our brain that are like hierarchically more superior than the cortex. So when you make a movement and you know, you say, oh, I did it consciously, you did. But there were other structures that actually dictated your cortex how to do it. We're going to talk about it.
Okay.
So far, am I making sense? I want to highlight one important thing, folks. And I want you to, I'm going to repeat those words again and again. Central nervous system, whether it's brain, brainstem, or spinal cord, does not contain nerves. Nerves are peripheral structures. Now, you may wonder how the different parts of the brain and central nervous system in general, how do they communicate? There are ways to communicate, but these pathways are called tracts rather than nerves. Okay, they functionally similar, they structurally different. T-R-A-C-T-S, tract, yes. So how do we know what different parts of the brain do? Well, originally the studies of the brain function and functional anatomy were focused on so-called loss of function approach. I found a really good analogy for this. If I tell you that you all will have to lose one finger, I know exactly which finger this is going to be. It's going to be the pinky on your non-dominant hand. That's it. Okay? Because you know that losing it, it's not such a big of a deal, but if I will try to chop off your thumb, that's the most important finger, right? So, scientists looked at people with various types of brain damage to understand, like, if we cut off this piece, what's going to happen, you know? Now we don't do that because it's not super reliable. We have better tools, and the most important tool right here is functional MRI. So when your brain is, when a part of your brain is real active, when the part of the brain is very active, it requires much more resources. Resources that are brought to that part of the brain by blood. So the active parts, more active parts of the brain, will have greater blood flow, and this blood flow can be detected in real time by means of functional MRI. So right now, for example, the parts of the brain that are responsible for hearing and understanding speech, parts of the brain, frontal lobe probably for, you know, analysis of the information, and the motor parts of the brain that control your writing muscles. So what we're going to do, we're going to discuss different cortices today, and we're pretty much going to call it a day for today. So I want to highlight something. Cortices are divided into three main categories. First category is motor cortices. They control movement. Movement, yes. And not just movement, specifically the movement that's produced, that your skeletal muscles produce. Okay? Then we have sensory cortices. They are responsible for conscious appreciation of sensations. And then we have association cortices. They're responsible for higher cognitive abilities. So we're going to start. So what do you need to know about the cortices? I'm going to give you an idea what questions you may expect. I may directly ask you to match the name of the cortex and its function. Okay? I may ask you to identify what a dysfunction of a particular cortex is going to look like. I'll give you examples, don't worry. And I will 100% ask you about the location of cortices. So let's start with the motor cortex, and I will provide you with an example of a question that you can see in the exam. Okay? So we're going to start with first of all motor cortices, no exception, are in the frontal lobe. Okay? They kind of a bit of in the back. You can see the motor cortices here. Okay? But still they are in the frontal cortex. So primary motor cortex sends signals to your skeletal muscles to produce precise and skilled movements. Okay? So, right now, when you are writing, are you using muscles in your arm and hand? Yes. Is your writing legible? Is it readable? For you, okay. My point is that these movements are coordinated. Okay? They are. That's your primary motor cortex. Okay? Now let's talk about the dysfunction. One of the most common types of a stroke is the occlusion, so-called ischemic stroke, the occlusion of a middle cerebral artery or any of its branches. Middle cerebral artery and its branches provide blood supply to motor cortices, pretty much. So the occlusion and subsequent death of brain tissue will result in your inability to send those signals to your muscles. Okay, and it will result in the contralateral paralysis. We're going to talk why it is contralateral eventually. Okay? Does that make sense to you? Now, here's my question. So imagine you have a patient with that classic damage to the primary motor cortex only. Patient is otherwise lucid. Say it's a left middle cerebral artery, so right side is affected. This patient cannot write. Does that patient know how to write? Do they still have a skill? Yes, they just cannot produce the movement. The communication between the cortex and the spinal cord is interrupted because there is no primary cortex. Okay? Now, unlike primary motor cortex, premotor cortex, which is also located in the frontal lobe, is concerned with the generation with planning of movements. Okay? It is a planning of movements. So it will receive, let me give an example. Okay? So let's say I want to jump on that table. Okay? So if I'm going to try to jump on the table while sitting in a chair, tough luck. I'm probably going to fail, right? Let's say I'm standing, okay? So in order to plan this jump, I need to receive a bunch of sensory information. Where's the table? How far is it from me? My body, in what position it is in? Am I squatting down? Am I standing up? Am I standing on something else? Premotor cortex will also receive inputs from basal nuclei that I already mentioned, and cerebellum. They will generate they will guide premotor cortex on how to execute the movement. And then premotor cortex will form this output signal, and importantly will relay it to primary motor cortex. So premotor cortex gives instructions, tells primary motor cortex, hey, communicate there, contract this muscle, contract that muscle. Essentially, your skills, what we call motor skills, are stored in your premotor cortex. So here's my question to exemplify this. Question number 1. You all know how to write, okay? If I will switch you for, I don't know, 10 years, only, typing, just no writing at all. 10 years later, I'm giving you a piece of paper, and I tell you to write something. Can you? Yes. You're probably going to be rusty, but you can write. So it's a skill. It's like playing piano, riding a bike. So imagine somebody has a brain damage that affects exclusively premotor cortex. Question number 1. Can they move muscles? Yes. Can they skillfully move muscles? No, they're going to lose some skills. It's been shown it can be retrained. There is some limited ability of other cortical parts to take over, but that's the dysfunction of premotor cortex. Losing the skill, but not the ability to contract. Pretty straightforward. Broca's area and frontal eye field. Frontal eye field is self-explanatory, I presume. It, just, controls your eye movements, that's it. Broca's area controls muscles of the speech, not your language. When people have a stroke, they get paralysis. Often they lose the ability to move the eyes, one specific eye, and they also develop slurred speech. Okay? That slurred speech is because of the damage to the Broca's area. They can't precisely move their lips, their tongue, whatever, to make legible words. If you give them paper and they can write, they will write. They find a language. Yes, go ahead.
Are you speaking like when they're having the stroke or after?
When they are having the stroke, if dying neurons are in the Broca's area, they will develop slurred speech. They still possess all the language capacity, they just can't move the lips and the tongue. Does that make sense? Okay? So, these are effectively the examples of the questions I'm asking. Like, what is the function of such and such cortex? And you tell me. Or, patient has slurred speech but maintains the language knowledge. What is likely affected? Broca's area. Okay? And where they are located, that is true for all cortices. So let's get to sensory cortices, and I want to ask you a question. What senses can you name? Huh? Well, go on. Touch, taste, smell, hearing, and sight. Yes. So, touch and everything that is somehow related to touch. is called somatic sensations. So somatic sensations come from somatosensory receptors located in the body, and the proprioceptors located in the muscles, joints, and tendons. Okay? So let me give an example. This, touching my fingers, is that a touch? Yes. How would you characterize, like, this? It's a pressure, right? It's a pressure, right? I'm pressing, right? I'm pressing. It hurts. So when it hurts, what do we call it? Pain. So touch, pressure, pain. When, like this, vibration, right? If when you touch ice, do you know that it's cold? Temperature. It all falls into somatic sensations. If you right now have a Charlie horse in your calf, do you know that you're having a Charlie horse? Yeah. You don't have to look at your calf. So muscle contraction or stretch, that's a somatic sensation. Now, if hypothetically some magician will start bending my knees without touching me, will I be able to tell the position of my knee, position of my joint, that it's bent? Yes. So stretch of a tendon, stretch of a ligament, position of a joint, contraction of a muscle, pressure, touch, vibration, pain, temperature, all of this are somatic sensations because they come from your body, whether those are structures deep or structures that are superficial, they are from the body. Primary somatosensory cortex, which is located in the parietal lobe on each hemisphere, is responsible for receiving the signals, okay, and interpreting basic qualities. Where? What is it? Is it a pain? You know, how intense? But when by using your somatic sensations, with your eyes closed, you can still recognize objects. Make sense? Like you don't really need to like see things sometimes. Like you fix something and you can't see, but you know what it's got to feel like. You know, oh, this is a bolt that I need to remove, or you in the darkness, you know, like you have to go pee and you touch the walls to figure out where the door is. That's your somatic sensations being integrated in the association cortex. So primary somatosensory cortex receives and kind of sorts out everything, and then association cortex puts it all together and tells you what this is. And as always, we're going to go into the dysfunction. So, go ahead.
So somatosensory associations that only tells us what things are based on the way they feel.
Absolutely. Only on somatic sensations. So, I'm going to give you an example. Imagine I lose a part of my primary somatosensory cortex that's responsible for receiving signals from my fingers. Can I feel things with my fingers then? No. I lose sensation, that's it. Not because my fingers are chopped off, not because signal doesn't reach, but because my primary somatosensory cortex just, nope, I'm not working. I cannot discriminate anything, okay? Now, it's more interesting if the function of the association somatosensory cortex is lost. For example, if you will reach into your pocket, and you have like some small objects in there, would you be able to tell like a board game chip from a coin and from a crumbled receipt from a grocery store without looking at them? Yeah, without looking, because your fingers will provide all necessary sensations. Those sensations will be put together, in the association somatosensory cortex. Am I clear? So when it is damaged, you take those, you still feel them, you just can't tell what they are, those objects. You have to take them out and look at them. You lose the ability to integrate the impulses. And this brings us to one of my favorite conversations, the conversation about somatotopy. So I want you to take a look at this map. On the left, you see the map of a motor cortex. Okay? You can see that different regions of the motor cortex, this is the cross-section, coronal section of the brain. So like you're looking like front, for example. Okay? And on the right, you can see the regions that receive signals from different body parts. So let's start with the motor part. So here, that part controls the muscles of your trunk. Okay? This part, like all of it, controls your fingers and thumb. I'm going to ask you a stupid question. In terms of the size of the muscles that move your trunk versus muscles that move your fingers, which muscles are, well, bigger? Trunk. And yet, your trunk muscles are controlled by a much smaller region of the motor cortex compared to your fingers. Why? Yes, exactly, because the movements that you can produce with your fingers are more complex. And I don't want to say that this is like, this is why. It's actually quite an opposite. You can produce really complex movements with your fingers because a greater chunk of the motor cortex is quote-unquote dedicated to their control. Okay? You can make fine facial expressions because the greater chunk of the motor cortex, like here, all of this is your face. Okay? Greater chunk of a motor cortex is dedicated to the control of facial muscles. Okay? The map? Of course. Dude. Seriously? Of course not. No. No, God forbid. No, you don't need to remember. I use it to illustrate the idea. So all in all, the size of the area that controls certain group of muscles corresponds to the precision of movements that these muscles can produce. Now, what about sensory receptors? Again, this is trunk. Very small area. Okay? This is your hand and fingers. It's a lot. While hand and fingers are much smaller than your trunk, they have a greater, the area that receives signals from your fingers and your hand in the cortex is much greater than the area that receives sensory signals from your trunk. Well, if it's not the size, then what? What is it? If it's not the size, so the part of the motor cortex that controls the body part does not depend on the size of that body part. It depends on something else. How sensitive, but what determines how sensitive your body part is? Not quite nerves. The stimulus, any kind of stimulus that generates nerve signal. What does it have to act on? The R word, the receptor. So different body parts have different number, different density of receptors. Make sense? You can actually do a very neat experiment. You can do it the hard way or the easy way. The hard way is more fun. It's called, I think we may actually do it later in semester. Spatial discrimination test, or 2-point discrimination test. You can do it at home. All you need is a blindfold, a ruler, and 2 pins or needles. So you put 2 pins or 2 needles. You touch the skin of the person. You need second person. You cannot do it on yourself. Okay. So you put those 2 pins on the person's skin and ask them if they feel 2 pins. And they say, yeah, I feel 2 pins. You blindfold them. You move the pin, 1 pin, closer to another. So you shorten the distance between them and ask them, do you still feel 2? And they say, yeah. And you keep moving them closer and closer until they answer to your question, do you still feel 2 pins? They say no. There will be distance between the pins. There will be. But the distance will be much smaller on the most sensitive regions and tremendously large on less sensitive. So the distance that you will get to on the hand or the finger or neck is extremely sensitive, especially the back of it. We're talking 3 to 5 millimeters between the pins. Trunk or like thigh, centimeters. It's really fun. How to do it the easy way? If you have access to a fairly gullible child, tell them to close their eyes and extend their arm, and tell them to not open their eyes until you, when your finger is going to reach their elbow, the antecubital region. So you start at the wrist and you slowly move your finger towards the elbow. And they will stop you. And if you play smart and change the speed at which you move the finger, they will never, ever be able to stop you exactly in the elbow. Because we cannot discriminate at some point between this and the elbow if our eyes are closed. We played it elementary and middle school. Okay? So it's all about the number of receptors, not the body size. So far we covered touch and all associated. What about other senses? So visual cortices. Visual cortices are located into the occipital lobe of the brain. And just like somatosensory, are divided into primary and association cortices. So primary cortex receives, whatever retina of your eye sends to the brain, color, movement, contrast, shape. But then association cortex, it interprets those signals and allows us to recognize things. Am I clear? Now, in full transparency, okay, it's actually very interesting to talk about this. So first of all, there were things that you saw for the first time, truly. And some of those things, you had no idea what they are. Like, okay, right, you know that, okay, this is the building. But there was a time in your life when you were very little. You didn't know that this is a building. Like, what is that? What do people do with that? Okay? So somebody told you this is the building. And then your cognitive power realized, oh, if it looks like they're different buildings, but you can tell, oh, like this is a typical building. But you can imagine some crazy architect build some outrageous structure, which effectively is a building, but looks like a giant mushroom, something like that. Okay, I don't know what it is, right? So your recognition of things like, okay, I saw that, this is the cabinet, okay? It comes from both visual association cortex and your memory. You need to remember. Your emotional response to what you see comes from a different part of your brain. So far, do you understand the difference between the primary and association? Primary cortex, if it's damaged, you can't see. Association cortex, you can see, you cannot recognize. You cannot put things together. Okay? Auditory cortex, located in the temporal lobe, and also is divided into primary and association cortices. Okay? Same idea. Every sound has three main qualities. It's a volume, it's a pitch, and its location. Your primary auditory cortex interprets where it comes from, how loud it is, and what pitch it has. Your association cortex recognizes the patterns of the sounds and can if you are on the phone with someone, you can recognize this person's voice. You can recognize, I can't, but people can recognize the sounds that birds make. Sure, that's your association cortex. Now, what about this? So olfactory cortex, or rhinencephalon, is also in temporal lobe, but it's one of my favorite cortices. Smell is probably my favorite sense to talk about eventually. But it's kind of diffuse. There are several structures that are very loosely connected to each other, but connected nevertheless, that form that cortex. Okay? Obviously, it is responsible for smell. Gustatory and visceral cortices are both located in the insular lobe. Okay? Gustatory cortex responsible for taste. Insular lobe is responsible for signals from the internal organs. I don't remember if I mentioned that, but the term viscera refers to all internal organs except for bones and muscles. Okay? And the brain. Are we good so far? So before we moving to association, multimodal association cortices, I want to give you some kind of highlights, ideas. If you look at what we've talked about so far, somatosensory, visual, and auditory cortices, each of them is divided into primary and association parts. Okay? So let me ask you this question. If I will blindfold you and ask you to recognize, say, your kids by touching their face, would you be able to do that? Probably yes. Because your somatic sensations, your touch, is very discriminatory because of that association component. Your vision is really good. Okay? Your hearing, human hearing, is probably second to whales only. We have fantastic hearing capacities. And yet, our taste and our smell aren't that great. Okay? And it's like, think about this. I'm going to give you some examples. You probably won't be able to recognize, and I know it's going to sound a little off-putting, but if 3 different people with 3 different diets peed on the wall, you won't be able to tell apart who peed what, right? Or if you would read the stories about people doing blind whiskey tasting and not being able to recognize different kinds. Our taste and our smell aren't that great if we compare it to other animals. Hearing, vision, and touch are pretty good. I know this is a fairly speculative idea, but I have an idea why this is so. Humans. Where did humans originate? Where the first Homo erectus, upright. Yeah, we started in Africa. How did we get food? Oh, not hunting, no. Africa is full of very good hunters. Lions, leopards, other predators. If not hunting, gathering, yes. Yes, we were scavengers. What is the best time to scavenge food after those lions and leopards? Day. It's a day because they're active mostly in the evening or in the morning. So we were daytime scavengers and gatherers. You need really good vision to successfully spot the carcass, and you need a really good touch to produce instruments that will help you to clean the meat off of the carcass. And you need really good vision and hearing to not be eaten in case something dangerous is around. If you're a scavenger, having a really good sense of smell may be a disadvantage, because this shit can stink, and the taste may not be great. Although if you think about the dogs, I think ours enjoys smelling poop the most. I don't understand that. Okay? So now we're going to shift to multimodal association cortices, and they are effectively what makes you you. So, anterior association or prefrontal cortex contains all your personal distinctive features. The way you learn, your demeanor to other people, everything about you, your personality. And the damage to that part of the brain changes the personality. Okay? So the reason why we know that, there's a plenty of evidence. My favorite story, you probably know that. There was a worker in England, was working in the docks, and a metal rod pierced his frontal lobe, specifically prefrontal cortex. Interestingly enough, he survived. He survived the damage. They removed the rod. He changed from very polite and almost timid worker to aggressively loud and profane asshole, pretty much. Okay? So you probably have heard about lobotomies. What were they used for? What was the purpose of lobotomies when they were used as a medical procedure? Yeah, basically to treat potential neuropsychiatric disorders. Okay? And if you forget about all the, moral restrictions and ethical considerations, and just look if those disorders went away, they did after lobotomy, along with a bunch of other stuff. Okay? It's like treating, an athlete's foot by amputating the entire leg. Okay, technically it's gone. And in addition, lobotomy changed the personality of people. So it wasn't like if I get my leg amputated, overall, I'm kind of going to be the same. Like if I'm sick and I'm told, the only way to survive is to cut off your leg, I'm going to be traumatized, but overall, I'm going to be the same person. The person after lobotomy is not the same person. Wasn't. Okay, we don't do it anymore. Does that make sense? Now, one important feature of that cortex is that social environment really important for the brain development. So there were studies showing that actual anatomical structure of prefrontal cortex in people who were raised by animals, and there are examples, differs from the people who were raised by humans in a proper social context. And this change, this alteration, this deficit, it's permanent. Because brain pretty much forms by the age of 18, 20, maybe 21. So the lack of social interaction early in life, it's very detrimental. And by social interaction, I'm not saying that, you have to take a baby to a nightclub or something like that. But even parental interaction, talking, playing, communicating, in other words, is critical. Posterior association cortex. Specifically, I want you to know the function of the Wernicke's area. To my knowledge, Wernicke's area is, in humans, is the largest among all mammals. The largest in all, mammals or animals, obviously, per brain size. Wernicke's area. Wernicke's area, this one, is responsible for your language abilities. Does that make sense? Okay. So, humans are pretty much the only species that have very complex language. And I mean very complex. So, it was a mutual thing. Think about this. Okay, we're going to go back to evolution. Let me just say a few words about limbic system, but I want to talk about, the brain development and human evolution a little bit. So, limbic association area, or limbic system, located mostly in the temporal area. I'm sorry. This is your conscious emotions, let's call them this way. So, imagine that you are seeing someone, like you running into someone who you like a lot. Okay, like seriously a lot. You're going to be happy. Yeah. Would you understand that you're happy? Also, yes. Or your boss is being an absolute jerk and you want to punch him in the face. But you don't, despite the fact that you're really enraged, but you understand this rage. If I will punch that person in the face, I will get fired and I can't afford right now to be fired. So we're going to talk more about limbic system. It's not only your conscious understanding of emotions, it's also a bunch of your emotional, physiological responses to emotions. Okay? Does that make sense? Now, I want to kind of go back to the brain development and say a few words about that. First, let's start with the human evolution, all right? Humans are the most advanced social animals. Our social structure is very complicated. It can be complicated because we can talk. And early, if you think about this, the better humans of a particular tribe, I don't know, genetic background, the better they could communicate, the more chances they had to survive. If they survived, they left the offspring, so classic evolutionary pressure, okay? Now, do we evolve now? Sure. Probably de-evolve, but that's beyond the point. But so this shows you that overall, people with larger Meninges area with greater capacity for languages often had evolutionary advantage, one way or another. Now, that brings me to a very important question for you. People, when they born and they grow up, their brains, do they all develop in the same way? No. Well, first of all, it depends on what they're engaged into, right? Like, when I was a kid, I was chubby and clumsy, and I never played really any team sport. I'm not great with neuromuscular coordination still. Now, I have twins, and they both played soccer. One is way more coordinated than another. Like, same age, everything. One is more coordinated. There are intrinsic better or worse abilities. Okay? If somebody has an absolute pitch, the person can just repeat the musical theme just by hearing it. That's developmental and genetic advantage. I played musical instruments when I was growing up. I don't have an absolute pitch. I played a lot, but I don't because I'm not that genetic. Same is with Meninges area. Some people have better skills for languages. There was a story, I don't remember which country it was. I find it pretty funny. So they were about to put a guy to death, really 16th or 17th century, and they needed to give him whatever religious thing that you give to person you're about to kill, so the God will not be mad, right? Whatever, last rites of animal. So the dude, nobody spoke the language that the dude spoke, but they had a guy, some priest, who was really good with languages. By that time, he spoke like 25. He learned that language overnight. So yeah, probably he had like gigantic Meninges area, and that plays into child development. If you start playing, I don't know, soccer with your child since day 3, they're going to become really good. But they're going to be very coordinated and good at soccer. If a child grows in multilingual environment and they learn multiple languages, it's going to be easier for them to learn languages afterwards. We do have that experience with our kids. And we do have the opposite situation. One of them is real good at languages. Another one is fine, but not outstanding, and probably just because they grew up in bilingual environment. If they are what?
Like if they are raised in a language household.
Overall, there is a very strong correlation between growing up in the bilingual or multilingual environment and general cognitive abilities. And there is a very strong correlation between coordinated physical activities, be it tennis or basketball, and cognitive abilities as well. Okay? Yes, there is. But you have to remember, there are just genetically, there are preferences. Some people like some kids like to read and other kids like to play, and it's totally fine. And we see this all the time, in our kids, because they now pretty much adults. So the last thing to talk about is lateralization of functions in the cortex. So you can see that different tasks are dominated by different hemispheres. Now, what does that mean, dominated? So if you look at this image, verbal memory points at the left hemisphere. Can you see that? Does that mean that it's only left hemisphere? It doesn't. It means that the dominant structure that is used first, so to say, for verbal memory is in the left hemisphere. But if something happens to it, right hemisphere is going to take over. Okay? We do have lateralization of the functions in the way that your left hemisphere controls, for the most, your right side of the body receives sensory inputs and controls muscle movements, and right hemisphere does the same to the left. So cerebral dominance is determined by the hemisphere that's dominant for the language skills. If you start looking at this image and you think, in somebody who is left hemisphere dominant, there will be more rational, and in somebody who is the right hemisphere dominant, there will be more musical. More and more data show that when we assign the dominance based on being righty or being lefty, it's all bullshit. So the idea that left-handed people are very artistic and right-handed people are very logical and rational, data do not support it. What data do support is the story about ambidextrous people. You know what ambidextrous people are. They can use both hands and feet equally well. They don't have it. Like I had a buddy when I was a kid, when I was in middle school. We played table tennis, ping pong, and he was ambidextrous. It was insane. Like he could switch hands while playing. Crazy. But, say again? Yeah, he could use both of his hands for things like playing or eating equally well. People who are ambidextrous statistically have much greater risk of psychiatric disorders. Potentially because there's it's not normal. Your wiring should be kind of not that. There should be some dominance. Okay? So we're going to stop here.